So today's topic is going to be photosynthesis. We're going to talk about light reactions, dark reactions, and a little bit more detail than we did when you were in ninth grade. So first we're going to talk about the evolutionary significance of photosynthesis. So in general, we know that photosynthesis is an organism's ability to capture and then store energy that's going to be used in biological processes. Okay, and in this particular case with photosynthesis, we're capturing energy from the sun and we're producing sugars. That's the stored form of the energy. So the organism that's doing photosynthesis is not creating any energy, right? They're just doing an energy transfer, following that first law of thermodynamics. They're capturing the energy from the sun and they're transferring it over to the chemical energy that we would find in the sugars. So photosynthesis actually first started in prokaryotes. So we say that photosynthesis occurs in a chloroplast, but remember a chloroplast is a membrane-bound organelle. So in a eukaryotic cell, it will occur in a chloroplast, but prokaryotic cells can also still do photosynthesis. They have certain modifications that allow that to happen. So your prokaryotic cyanobacteria were the first ones that were doing photosynthesis. And we have evidence to show that this is what has helped create our atmosphere that contains oxygen, right? The byproduct of a lot of these cyanobacteria doing photosynthesis helped increase the oxygen levels in our atmosphere enough to make it really so that organisms could live on land. The oxygen in our atmosphere helps to regulate the temperature on land, and so it made it so that organisms would be able to move and to live on land. You can see in this picture over on the side of this cyanobacteria here. Okay, it's got its genetic material, it's got its ribosomes, and it also has a thylakoid membrane kind of built in right here, kind of built in along the edge of its cell membrane. So it doesn't have separate enclosed thylakoid membrane areas in a chloroplast, but it does still have the extra membrane that allows it to do photosynthesis. The membrane is required. There has to be some sort of compartmentalization to do photosynthesis, and you can see where that's happening with these various membranes here in this prokaryotic cell. So eukaryotic photosynthesis is based on these prokaryotic pathways. Remember our endosymbiotic theory? That a smaller prokaryote was engulfed by a larger prokaryote, and that eventually over time they couldn't live separately, and that's what led to eukaryotic cells. And this would be an example of a smaller prokaryote being engulfed by a larger one, and of some form of this cyanobacteria is what would have eventually become our chloroplast. So if you remember, the photosynthesis is made of two basic types of reactions. We have our light-dependent reactions, and then we have the Calvin cycle. Both of these are going to be controlled by enzymes, right? All of the reactions that occur within the light-dependent reactions are going to be regulated by enzymes. Enzymes, remember, control all of our metabolic pathways. Sometimes you'll see these things referred to as metabolic pathways, and that's what this is, right? I'm transferring energy, so it's a part of my metabolic pathway. So the light-dependent reactions, they're going to occur on the thylakoid membrane, the chloroplast. If you follow your picture here, right, we've got a leaf, and then our next step here, we're looking at a cross-section of the leaf, and then we're zooming in to the individual cells in the leaf and zooming in on that chloroplast, and you can see all the little stacks of thylakoids in our chloroplast here. If you remember from freshman year, your diagram of your chloroplast, right? So this whole thing here is my chloroplast. The individual little stacks, each one is a thylakoid. Right? You have these, remember you can see the green, but they look kind of squished there. Right? You have the stacks of membranes there. Each individual one is a thylakoid, right? The whole stack, remember, was a grana. And the liquid part of the chloroplast that surrounds all the thylakoids, like you can see it's gray in this little diagram here, that liquid part there is the stroma. So the light-dependent reactions, they occur on that thylakoid membrane. And that thylakoid membrane is folded and stacked. You can see in this bottom section here how the membrane doubles back on itself, it's folded and it's stacked because it increases surface area. We've already talked about how increasing surface area can make a cell more efficient, in particular with, like we talked about surface area to volume ratio, bringing in nutrients. So the same thing is correct here with these light-dependent reactions. The more surface area I can create to trap light, because the thylakoid is where I'm going to find those chlorophyll pigments. So the more surface area I can create to trap light, then the more efficient I can be at this reaction. The more I can do of this reaction. Okay? So an increase in surface area is beneficial. And the folding of the membrane is what allows for that increase in surface area. So the main job of the light reactions is to actually capture the light energy. Okay? So they're trying to capture the energy that's in the light, and they're going to produce ATP and NADPH, both of which are needed to power the Calvin cycle. So if the light reactions aren't working, then the plant cell isn't producing ATP and NADPH for the Calvin cycle. Remember, a plant cell is going to have a separate mitochondria that's going to make ATP to power its other chemical reactions. But the ATP that's produced during the light reactions is specifically harnessed to finish powering photosynthesis. That way the cell doesn't have to get ATP from a whole different place. Okay? And that ATP, we'll talk about how it's created, is created in a similar manner to how it's created for cellular respiration. And so those two processes are going to be very, very similar. So like we mentioned, the light reactions are producing the raw materials for organic molecule production. Our organic molecule, remember, would be our carbohydrate. That would be our sugar that's being produced. And this diagram is similar, but now you can see where this ATP and NADPH are headed over to the Calvin cycle. And if you think to your summary of photosynthesis, right, the fact that it's carbon dioxide plus water is giving me oxygen and glucose, you'll notice ATP and NADPH are never mentioned. That's because they are recycled back and forth between the light reactions and the Calvin cycle. So I don't need an extra input of them, nor do they come all the way out of my reaction. So chlorophyll is key for the plant or the organism in general. Could be, again, a photosynthetic bacteria. Chlorophyll is key for it to be able to do photosynthesis. Chlorophyll is the green pigment. Remember, pigments are the dyes. They're going to help us. The pigment is the dye that's going to help absorb the light energy. Okay? So embedded in that thylakoid membrane, you're going to find a bunch of this chlorophyll. This picture over here on the left, you can see how the chlorophyll here is embedded in the membrane. And so that chlorophyll determines what light energy is being absorbed. You know that the light energy is available on the UV spectrum. Remember your Roy G. Biv, right? Your colors of the rainbow. So those colors come from different wavelengths of light. You can see in the graph down below here. So on the x-axis here, we have our wavelengths of light in nanometers. And so it shows you to which ones correspond with which colors correspond with which wavelengths of light. So plants absorb energy, the most common in the blue-green areas of the light spectrum. You can see we have two kinds of chlorophyll A and chlorophyll B. And you'll notice over here on the y-axis is the absorbance. So I'm absorbing a lot of light energy if I'm in a plant in this 400 to 500 range. Okay, the purple, the blue, just a touch of the green. This carotenoid here, this is a different pigment. It's more of a yellow, orangey colored pigment. We see more of it when the leaves change color. But you'll see it can still absorb light. It absorbs a little bit more of the green light. And then you see the chlorophyll A and B also absorbing some light down here around the 650-ish to 700 nanometers. So some of that orange and some of that red. And if you think back, remember we see the light that is reflected back to us. So that means all this light that's down low right here did not get absorbed. Well, if it didn't get absorbed, right, because its absorbance level is really, really low, if it didn't get absorbed, then it had to get bounced back to us. and you'll see the colors that are getting bounced back to us are those greens and yellows. So that's why plants look mostly green. So chlorophyll is the pigment that makes the plant look green because it bounces back a lot of green and yellow light. But it's really good at absorbing purple, blue, some oranges, some reds. So that energy that's absorbed from the light is used to excite electrons. That energy is used to basically power the ATP production of the light reactions. So let's look at how that happens. So embedded within the thylakoid membrane again, so you can see your diagram here of your chloroplast, zoomed in on a thylakoid membrane. So we've got these clusters of pigments. And within those, you're going to find what are called photosystems. And there are two photosystems, photosystem 1 and photosystem 2. And unfortunately, they're backwards in their order. They are named for the order in which they were discovered. So photosystem 1 was discovered first, and then photosystem 2. However, unfortunately, photosystem 2 is the one that the light hits first, and then the energy is transferred to photosystem 1. So they actually work in the reverse order. So this is where I'm going to find the highest concentrations of the chlorophyll. I'm going to find the highest concentrations of my light gathering molecules because this is where the plant wants to focus the light. We want to focus the light absorption here because we need to concentrate that energy. So over here on the right, you can see the light energy coming down from the sun, okay, being absorbed into the photosystem and then being passed from complex to complex. So that energy is going to get transferred between the photosystems. Really the difference between the photosystems is what wavelength of energy they absorb the best, which is why the photosystem 2 absorbs straight from the sun, and then as that wavelength of energy changes as it gets processed by the chloroplast, photosystem 1 can absorb that better. Okay, so let's get into some details here. Again, these details, some of them you've seen before, so let's really focus on the new parts where we're adding. So we're still concentrating here on the light reactions. So in our picture here, what we have zoomed in on, you'll see there's our stroma, which is the liquid part. Here's your chloroplast, right? The stroma is the lighter beige part there, the liquid part. The thylakoid is the darker kind of orangey peachy. You can see here's your thylakoid space, my thylakoid membrane. So hopefully we're oriented a little bit here. I've got my thylakoid space is physically separated from my stroma by the thylakoid membrane. And that's important. If we don't maintain that physical barrier, then photosynthesis can't happen because we can't create certain concentration gradients. So this is another, again, example of structure dictating function. If this structure is damaged and there's a hole in the thylakoid membrane, then I cannot create concentration gradients and photosynthesis will not occur. So we've got the light, the energy from the light is going to split the water molecule, and that is called remember photo is light, photolysis. So I take this energy from the light and I use that energy from the light to split the water molecule. That's a chemical reaction. It's going to be catalyzed by enzymes. Okay, and I'm going to split the water molecule. And I'm going to split the water molecule into hydrogens and oxygens. And you can see that happening right here at step 1 in our diagram. So the water has been split into oxygen molecules and it's been split into hydrogen molecules. And so these hydrogen molecules, remember hydrogen ions are H+, right? Hydrogen molecules have one proton, one electron, and I can strip that electron off and make it become a hydrogen ion relatively easy, right? A very brief chemistry review because I know some of y'all are in chemistry right now, right? We've got our one proton in the middle of this atom and one electron on the outside of the atom. So if I can strip this electron off, right, I can remove this electron. Now I have no electrons on the outside and just my one proton. So this becomes my ion, right? It has a 1 positive charge. So these electrons can be stripped off those hydrogen molecules. And now I'm left over, you can see it right here, with 2 hydrogen ions. And the electrons are transferred to these proteins. You see these very large purple structures embedded there in your thylakoid membrane? Those are proteins. What I have here is a chain of proteins. It is an electron transport chain. We talked about electron transport chains your freshman year. Remember, they pass the electrons down through the proteins, and that helps transfer energy. So your photosynthesis has an electron transport chain, just like you saw in respiration. Remember, respiration is where we talked about electron transport chain before. So the light energy is used to split the water molecules. The oxygen is released, right? This is where the oxygen product is going to come from. Okay, so the oxygen is released. You don't need it later. So the oxygen will be released from the plant during the light reaction. And the hydrogen atoms will be stripped of their electrons to then become hydrogen ions. And so the light energy is then transferred to photosystem 2. You can see it happening right here. So the light energy is transferred to photosystem 2. And what that does is it transfers energy to electrons that are stored in photosystem 2. So you've got 2 little electrons that are stored here in photosystem 2. And when they get hit with the light energy, think of it like they get really excited and they jump up. They get a huge input of energy and they jump up and they leave. So now these 2 electrons have left. Well, now I have 2 big holes in this photosystem. I have 2 energy holes in this photosystem that I need to fill. That's why we're splitting the water. I can fill these holes with these electrons that came from splitting the water. So that's what it means here in this second bullet point right here, where it says light energy is transferred from the pigment molecule to photosystem 2, where the electrons get excited. So the light energy hits photosystem 2. The electrons there get all excited and they jump up, okay, and they leave. And so they get so excited that they leave, but now I have to replace them. And so I replace them with the electrons that were pulled from the hydrogen. So these are my replacement electrons that go into photosystem 2. And now those excited electrons, the ones that got originally hit with the light energy and got so excited they jumped up, they now can travel down this electron transport chain. I know this diagram's gotten a little bit messy. We're going to look at it again on our next slide. Okay, so we have our same diagram here. This one's nice and clean now, and we're moving forward with what's happening. Okay, so far I've had the light energy hit the photosystem 2. Those electrons got super excited because they got full of energy, and so they jumped up and left. So those electrons are now traveling down the electron transport chain. So now I have to replace those electrons, and those electrons got replaced by the electrons that were on the hydrogen molecules that got separated, that got released when water was split apart, also using the light energy. So I've got these electrons moving down the electron transport chain. And you'll notice that your yellow arrows, moving those electrons, the yellow arrows on your diagram here are your electrons traveling down your electron transport chain. And you'll see right here in the middle, you don't really need to remember all the names on this electron transport chain, but you see right there in the middle, you've got 2 hydrogen ions that are also getting pumped across. So I have 2 hydrogen ions from when I split water. I'm also getting hydrogen ions being pumped across as those electrons move down. Remember, as those electrons move down the electron transport chain, that is transferring energy. And so the thylakoid membrane, the proteins there, can use that energy to pump hydrogen across. So I'm creating a proton gradient. Remember, a gradient is when I have differences in something, and in this case, I'm going to have differences in protein concentration. So what I'm doing is I'm building up protons in the thylakoid space. I'm building up a positive charge, right? Protons are 1 positive charge ion. So I'm building up hydrogens. Proton gradient is the same thing as a hydrogen gradient. I'm building up hydrogens in the thylakoid space. When I do that, those hydrogens naturally want to diffuse out, right? We need to be thinking cell transport stuff right now, right? I used energy to actively pump it over to force the formation of this hydrogen gradient. A whole bunch of positives don't want to be located in the same place, so they want to be able to leave. But they only have one exit door. And that exit door is over here. It's ATP synthase. And so when those hydrogens flow through that ATP synthase, that will naturally occur. It won't require any energy because I've built up and formed that proton gradient, so they want to escape. And they will escape through that ATP synthase. And when they go through, you can see this little hydrogen ion here going through that ATP synthase. When they go through that ATP synthase, that will phosphorylate ADP into an ATP. Phosphorylation, I just added a phosphate to that ADP. So it went from adenosine diphosphate with just 2 little phosphates to adenosine triphosphate with 3 phosphates. So I added a phosphate there, and now I have ATP that I need for the Calvin cycle. So the movement of those electrons down that electron transport chain is helping to create ATP because it's creating the proton gradient, which allows the hydrogens to then flow through the ATP synthase to create the ATP. That hydrogen flow is called chemiosmosis. Okay, so that hydrogen diffusion through the ATP synthase because of the proton gradient is called chemiosmosis. Those electrons then, you'll notice now they've ended up in photosystem 1. Photosystem 1 also absorbs light energy. So remember what happens when electrons absorb light energy. They get really excited and they jump up. So these electrons in photosystem 1 get really excited and they start to flow down another electron transport chain to their final electron acceptor. Their final electron acceptor here is that NADP+. So electrons are negative, right? So they're going to be attracted to that positive charge on the NADP+. So those electrons will flow down to that NADP+, and that will form the NADPH. Right? I can add 1 hydrogen, right? If I add a hydrogen, now if I just add a hydrogen ion, I'm at NADPH++, right? So I need 2 negatives to counteract those 2 positives, and that would be my pair of electrons, right? So I add my pair of electrons, the hydrogen ion, and now I get NADPH. Okay, and so that is my 2 products that I need to run the Calvin cycle, NADPH and ATP. Okay, and it's all due to the light absorption here. Photosystem 1 down on the farther end, right? Its electrons got excited, traveled down to make NADPH. Okay, and its electrons had to get replaced, just like the ones from photosystem 2 when they got excited and left. But this time they're replaced from the electrons that headed down the chain from photosystem 2. So it's just a constant chain moving the electrons that contain the energy from photosystem 2 to photosystem 1, finally to NADPH. And as they move down the chain from photosystem 2 to 1, they'll produce ATP. One of the things we need to realize, though, is that the plant will produce more ATP or more NADPH than ATP. Not really that it produces more, that there is a increased requirement. The plant needs, let's reword that better. The plant needs more ATP to do photosynthesis than it needs. Let me clear this off. Okay. All right, so now I have more space here. The plant needs more ATP to do photosynthesis than it does NADPH. So sometimes your plant will end up with like a backlog of NADPH. So lots of NADPH, but not enough ATP to run the Calvin cycle. And so the electrons then will start to do what is called cyclic flow. And when they do this cyclic flow, the electrons, instead of going from photosystem 2 to photosystem 1 to NADPH, will go from photosystem 2 to photosystem 1 and then circle back to photosystem 2. That way they just keep flowing through this section right here. And this section from photosystem 2 to 1 is the one that helps make the ATP. So if you hear or see the term cyclic flow, this is a cycle, right? It's a circle. And that means that the plant is making the extra ATP. The electrons are not going down here to make NADPH right now because it has plenty. And so they're just doing that cyclic flow and just going in a circle here, making the extra ATP that's needed to finish making photosynthesis happen. So now we've got the products that we need to make the Calvin cycle happen. We have this ATP and an ADPH. So now we actually have to use them to build the carbohydrates. So the Calvin cycle is occurring in the stroma, right? So outside of the thylakoid membranes. So it occurs in the stroma. It's going to build the carbohydrates. This is where the plant's going to actually need the carbon dioxide now. So the carbon dioxide is going to be used to provide the carbon for the glucose. The energy that's used to rearrange the carbon molecules, because I have carbon, I just need to rearrange it into a sugar molecule. So the energy that is used to rearrange those carbon molecules is going to come in the form of that ATP from the light reactions, okay, as well as the NADPH from the light reactions as well. We will kind of draw out a quick cycle of this, kind of showing how these electrons get rearranged. We will do it in class though, where it will be a little less messier than the video. But basically what will happen is the plant has some basic carbon molecules already in it, but those carbon molecules need to be rearranged into an actual sugar molecule. And so the ATP and the NADPH are going to they're going to provide the energy that allows that rearrangement to happen. So that's what this cycle is right here. The actual Calvin cycle is it's a constant rearranging of the carbon molecules, which will allow a sugar molecule to pop out on occasion. And then it'll rearrange the molecules again. And when the carbon is inputted, it keeps rearranging them into a sugar. And then it just continues on and on. And like I said, we will sketch this out in class where I think it will make more sense. And it will be less messy because I'm not drawing on an iPad with my finger. And so as we wrap things up, don't lose sight of your big picture, right? Your big basic picture of photosynthesis. Carbon goes in, hydrogen goes in, we need some light energy. Glucose comes out, oxygen comes out. This oxygen came out during the light reactions. Glucose came out during the Calvin cycle. Water was needed for the light reactions. Carbon dioxide was needed for the Calvin cycle. But again, do not lose sight of the big picture. My overall reactants, right? Remember, this is what goes into a chemical reaction. So my overall reactants, my overall products, and the fact that all of these energy conversions and breaking things down, I'm going to have enzymes. I'm going to have enzymes that are going to help regulate these processes as well. Okay, all right, so we'll do some labs with this. We'll go through the Calvin cycle diagram in class, okay, and we'll model this a little bit, and we'll get this all, settled into your brain for you.